Dr. Neu, to you. Joe is a longtime friend, colleague, fellow traveler. I'll tell you about that in a second. And he is our visiting professor for the next few days here. He's going to be speaking at the Neonatal Nutrition Conference that we have annually, which take place tomorrow and Friday. And today he is on campus. He'll be meeting with our fellows, and it's just great to have him here. He is probably the go-to person when you think about neonatology, microbiome, probiotics, and so on. He's actually an upper Midwesterner. He is kind of grew up in those around here, went to school in Wisconsin, went to medical school at University of Wisconsin, did his residency at Hopkins, and then went out to Stanford, did his neonatal fellowship there. Came back to the Midwest and was at Medical College of Wisconsin, and then, smart guy, moved to Florida and has stayed there ever since. So it's nice to have you back up here. Joe's work is really with one of the most devastating diseases that we see in neonatology, which is necrotizing enterocolitis. Much of his basic work has been on gut integrity. More recently, his work has been in the role of the microbiome, both acutely for the babies in the NICU, but also as it relates to long-term health outcomes. He was part of what is called the Pre-B group. This is a group that was put together by NIH and by A&D and the USDA to determine what are best practices for feeding neonatal intestines, if you will, or neonatal premature babies. And I encourage you to look at the article in the American Journal of Clinical Nutrition on that. He also wrote a seminal article in the New England Journal with Alan Walker, a name that should be familiar here, on necrotizing enterocolitis. I know Joe a little bit better from the fact we travel quite a bit together. Joe got involved with a group called Hippocrates. Hippocrates is a teaching organization that teaches neonatology to underserved and sometimes well-served areas around the world. He's now chairman of the board of that, and this is a group that goes around and talks not just about nutrition and GI issues, but pulmonary and ID and so on. Joe's published something like 300 plus papers, an astounding 122 articles, invited reviews, which really tells you he is the go-to person on this subject here. So he is here. He's going to talk about the microbiome during pregnancy and early postnatal life. And Joe, to have you here. Thanks.
Thank you, Mike. It's a real honor to be here. Being a Midwesterner, it was kind of interesting to get off the plane yesterday. Got off the plane and realized, hey, it's nice and cool in here. It's been in the 90s in Florida, so I think 40s and 50s is really good. I did bring a jacket along. So here's my disclosure slide, no conflicts of interest. And over the next 55 minutes or so, I'm going to talk a little bit about the Human Microbiome Project and intestinal microbe-host interactions. And then I'll talk about some recent studies as they pertain to preterm birth, mode of delivery effects, and environmental effects, such as antibiotics. And then I'll talk a little bit about microbes in human milk, and then the future, where we're going from here. So let's start with this slide. And if one of us went over to the Mississippi River or even to one of the more pristine lakes up in northern Minnesota and took a drop of water, put that under a microscope, what we would see is that there would be microbes. Put that under the high power, you would see a lot of microbes. But if you took that same drop of water and put it into a regular agar plate, you would be able to culture most of the microbes present in that particular sample. This is something that we've known about for over 100 years. And this is something called the Great Plate Count Anomaly. So this is the case in soil. Okay, if you take a soil sample and try to look at the bacteria in soil, you know that there are bacteria there, but you may not be able to culture them. Same thing holds true for certain niches in our body, such as the gastrointestinal tract, we are not able, with regular means, to culture all of the microbes in our gastrointestinal tract. So with the advent of the Human Genome Project, which started in the 1990s, a lot of new technologies were developed, and these technologies were applied to this area of the microbes that we are not able to detect very routinely using usual culture-based techniques. And two of the technologies that were developed are seen in this slide. One is called 16S rRNA sequencing. And what that does, it takes a 1,500 nucleobase region from the microbial genome, and there are about 8 of these regions that are highly variable and other regions that are highly constant. And you chop this up with enzymes, and with these enzymes you're able to take, you're able to find certain little sequences, and you put them next to one another, and you find these contigs and put them next to one another. You put this onto a computer, and it'll tell you what bacteria are present in a particular sample. So it gives you the names of bacteria. It gives you the taxonomy of the microbes present in that particular sample. It doesn't tell you much else. It can tell you diversity of the microbes. It can tell you which microbes are present in the highest abundance. So it does give some interesting information, but it doesn't give you the same kind of information that you would get with this other technology, which is called whole genome sequencing, or metagenomics. With whole genome sequencing, you get a better feel for all of the microbes that are present in a particular sample. You can name them, and you can also get a better feel with the newly developed bioinformatics, what these microbes actually do. Now, the whole genome sequencing is considerably more expensive than the 16S. 16S, you can now do a 96-well plate with less than $1,000, but it's a considerably more number of samples using whole genome sequencing. So these are the types of technologies that we have available to us now. And I'd like to begin this by also introducing you to a term that you may or may not have heard before. This is the term holobiont. What is a holobiont? It's a super or meta-organism containing an assemblage of different species that form ecological units. It adjusts and transforms itself according to environmental changes, causing evolution of the entire entity. And we are a holobiont. We as humans are a holobiont. Here's an example of a holobiont that's different than a human, obviously. This is a coral reef. And what is a coral reef made of? It's made of invertebrate animals. It's made of microbes. It's made of fungi. It's an ecosystem. And when this ecosystem is working well, when all of these different components of this ecosystem are working well together, you have this beautiful coral reef. However, with perturbations in this ecosystem, so for example, if the waters turn warmer with global warming, we're not supposed to say that in Florida, okay? Those are terms I can't use in Florida. But with global warming, okay, let's say that you start getting a little bit of a warmer ocean. There's more carbon dioxide that is present in that water. So there's more acid. And with that acidosis, the microbes change. And so you get this. This is bleaching. So this holobiont being messed up by something in its environment. And the same thing happens with us. We can have parts of our microbial ecology messed up by some of the things that we do, and we'll get into that shortly. Now, the gastrointestinal tract is really where a large number of our microbes are harbored. In the upper gastrointestinal tract, there are very few microbes. The stomach and the duodenum, if you take a sample of the contents, the luminal contents, it's about 10 to the 1. You go further down in the gastrointestinal tract to the ileum, it's about 10 to the 5th to 10 to the 6th microbes per milliliter of intestinal content. You get to the colon, it's about 10 to the 12th. So you have this abboral gradient of microbes that increase as you go further down in the gastrointestinal tract. They also play different roles in different parts of the gastrointestinal tract. So in the ileum, the major role that they play relates to the immune system. And in the colon, the major role that they play relates to something that we call a bioreactor role or metabolomic role. There is some overlap between the two, but in general, this is the case, that ileum is more of an immunologic role and the colon is more of a bioreactor role. Why? Because you have more tissue in the subepithelium of the ileum than you do in the colon. And the colon has a larger amount of bacteria, and we'll talk about that shortly. So here is the metabolomic or the bioreactor role. And at the top of this slide, you see this little insect. And in Florida, if we have a wooden home or any wood in our home, we have to be very concerned about this particular insect. It's called a termite. And termites have this unique capability to digest a very complex carbohydrate called wood. They have lignases in their gastrointestinal tract, and they have certain bacteria that are highly efficient in this digestive process. Now, in humans, certain humans have different microbial ecosystems in their gastrointestinal tract and their colons than others. And we are fat or skinny, as we see on the bottom part of this slide, depends on the types of microbes we have present. At least partially depends on the kinds of microbes present in our gastrointestinal tract. Remember, when we eat food, we are not just feeding our bodies, we are also feeding the massive numbers of microbes in our gastrointestinal tract, and these microbes produce a large number of metabolites. And they can also produce certain kinds of inflammatory mediators depending on the kinds of microbes present. One of my colleagues, Rob Knight, who is a bioinformatician who is now in San Diego, says that if he takes a stool sample from an individual who is obese versus an individual who is thin, he can generally tell who is the obese individual and who is the thin individual just by looking at the microbiome. So the kinds of microbes you see in the obese individual tend to be somewhat different. You have a lot more of a phylum called the Firmicutes phylum. The Firmicutes are able to break down certain carbohydrates and produce high levels of short-chain fatty acids, such as acetate, propionate, and butyrate. And these are then absorbed into the colon and utilized for energy production purposes. So you can see that there's a greater efficiency there with energy production purposes with different kinds of bacteria. If you take stool from an individual who is thin and put them into a germ-free animal early in that germ-free animal's life, nothing happens. That germ-free animal grows up to be normal. However, if you take microbes from an obese individual and put them into a germ-free animal early in life, that germ-free animal, as seen at the bottom of the slide, turns out to be an obese animal. And that ended up to actually helping us understand, or actually helping prove the concept that microbes play a role in terms of obesity. We had the data before that gave an association, but this actually helped at least partially fulfill Koch's postulates in terms of development of obesity and the mechanisms of obesity as they relate to the microbiome. Now let's go back in history, and here we have another insect called a Drosophila, which I'm sure you're aware of, and this lady her name is Christiane Nusslein-Volhard. Christiane Nusslein-Volhard won 50% of the Nobel Prize in 1995 for her work on Drosophila. In 1985, she was working on what causes Drosophila to differentiate into certain mutant types. There were certain mutant types that would grow their arms where the antennae should be, or their antennae where their arms should be. And so one of her graduate students was doing some protein gels one day and noticed that with the control, the wild type versus the mutant, there was a difference in the protein gel. She brought this gel to Professor Nusslein-Volhard, and Professor Nusslein-Volhard looked at that gel and said in German, "Das ist toll." Anybody speak German? Anybody know what toll means? Toll means cool, awesome. Okay. So this set of proteins got the name toll proteins. A few years later, this fellow, Charles Janeway at Yale University, was looking at macrophages and other immune cell types and found that there were certain proteins that interacted with components of the cell wall of certain bacteria. And he looked at these proteins and his laboratory characterized them and found that they were very similar in their characteristics as the proteins that Professor Nusslein-Volhard found. And he called them the Toll-like receptors. Now we know that there are about 12 of these Toll-like receptors, and these are very important in terms of our innate immunity. That's the immunity that we are born with. So, for example, Toll-like receptor 4 is very, very intensely being studied in various areas of inflammatory responses, including this disease that we see in neonatal intensive care, all too often called necrotizing enterocolitis. TLR4 responds to lipopolysaccharide, and if you have microbes, especially the gram-negative microbes such as Klebsiella, which have high levels of LPS, lipopolysaccharide, in their cell wall, you will have a high reactivity with these Toll-like receptors. These Toll-like receptors instigate a signaling response so that NF-kappa B and I-kappa B, which are cytoplasmic molecules, dissociate, and the NF-kappa B can then go into the nucleus and cause the formation of certain kinds of cytokines and chemokines, which are inflammatory mediators. So those are the Toll-like receptors, and in 2004, a very interesting study was published, also from Yale. At that time, Professor Janeway had already passed away, but his colleague, Professor Medjetov, was the principal investigator, the senior investigator of this particular study, which I think is very, very germane to us. The title of this is Recognition of Commensal Microflora by Toll-like Receptors is Required for Intestinal Homeostasis. They asked the question, is it important to have Toll-like receptors in the gastrointestinal tract? So I'm going to show you in the next slide a commentary from Jim Madera that was published in the New England Journal of Medicine shortly after this article came out in Cell. And so their hypothesis was as follows. Here you have Toll-like receptors, and here you have microbes in the gastrointestinal tract. And if you have these microbes and the Toll-like receptor is present, you have normal Toll-like receptor signaling, homeostasis of the intestinal surface, robust repair in response to injury. That's the normal, healthy situation. So what these investigators at Yale then did was they knocked out TLR4, TLR2. They did genetic knockouts of those two Toll-like receptors and also a linking molecule called MYD88. Okay, they had knockout animals of those three molecules, and they treated these animals and they compared them to wild-type animals with a substance called DSS. DSS is a very mild detergent-like material that can cause gut injury, but in low doses, it's very mild gut injury. So what they found, if with the knockout animals, that this DSS caused enhanced sensitivity to injury and diminished capacity to induce repair. They saw that by 1 week, these animals developed a hemorrhagic colitis and started dying after 1 week, just because they had this knockout and they were given this very mild detergent-like material. It hardly touched the control animals at all. Then they did something really interesting. They took the control animals, not the knockouts, and treated them with 4 different antibiotics where they wiped out the microbes, the bacteria, in the gastrointestinal tract. So here we wipe out of the microbes in the gastrointestinal tract, but normal Toll-like receptor signaling. And here's what they found. Within 1 week, those animals died of a hemorrhagic colitis, just like the knockout animals did. If they pre-treated those animals with small amounts of lipopolysaccharide or a Toll-like receptor 2 agonist, they were able to completely ameliorate this response. So pre-treatment actually prevents this, but wiping out all the microorganisms, and think about this, any of you who treat patients with antibiotics, antibiotics actually have this effect where you wipe out the microbes in the gastrointestinal tract, and you can actually cause some potential enhanced sensitivity in the gastrointestinal tract. We'll get back to that later. So the lesson from this article was low-grade stimulation or tickling of Toll receptors can prevent high-grade inflammation and intestinal damage and promotes intestinal homeostasis. Let's go on in history, and these three guys also won the Nobel Prize. Bruce Breitler, Jules Hoffman won 25%, and Ralph Steinman won 50% of the Nobel Prize. Well, Ralph Steinman, I want to mainly talk about his work because his work involved both the adaptive and the innate immune system, and he found a link between those two. This cell type right here is the cell type that he studied. It's called a dendritic cell. And in the gastrointestinal tract, we have dendritic cells. They're of the macrophage monocyte lineage, and they live underneath the intestinal epithelium, and they have this unique capability of sending out these dendritic processes into the lumen of the gastrointestinal tract, and they can take bacteria from the lumen or other antigens from the lumen, and they can bring them down to the subepithelium, and they also change the structure of their surface, the kinds of molecules that line their surface. With this change, they interact with undifferentiated lymphocytes in the subepithelium, and they cause these lymphocytes to differentiate into T effector cells or tolerizing T cells. Now, why is that important? Well, if you have an imbalance between the tolerizing T cells and the T effector cells, you can have disease. So, too many T effector cells, what happens is you can have these cells attacking one's own body. For example, type 1 diabetes, multiple sclerosis, all autoimmune diseases are caused by too many T effector cells. The tolerizing T cells, if you have too many of those, you might not be able to fight off infection quite as well. So you have to have a good balance. So the types of microbes that are present in the gastrointestinal tract act on these dendritic cells, which can help in the homeostasis of the T effector and the T tolerizing cells. So I'm going to start now with a little bit of the OB area and perinatology questions. Amniotic fluid is sterile, true or false? Placenta is sterile, true or false? Meconium is sterile, true or false? Human milk is sterile, true or false? Well, I know when Tom Pecora and I went to medical school, and Mike and I went to medical school, these were all true. I mean, this is, it's obvious that these were sterile areas. Well, we're beginning to question that, and I'm going to give you a little bit of background why we think that it might not be sterile. But I just want to point this article out to you. This was published in Nature just a few months ago, January 18th. And the title was Baby's First Bacteria. And this article really discusses some of the controversies between what is called the sterile womb hypothesis. And the author, I had the opportunity to talk to this author, Dr. Ms. Williard, and she calls this a scientific knife fight. And some of the people who believe that the in utero environment is still sterile considers the non-sterile environment studies fake news. So this is kind of an interesting area, and I think that I'm not going to say absolutely one way or the other, but I'll give you some of the information that suggests that perhaps it is not truly a sterile environment. So here is from a paper that was published from Stanford by Dan DiGuglio and colleagues, and Dr. Roberto Romero was a co-author on this study. Roberto Romero, who some of you may know, is the head of the perinatal institutes of the National Institutes of Health. And he has his laboratory in Detroit, and he has these samples of amniotic fluid in his freezers in Detroit that go back to the 1980s. And so, in collaboration with these people at Stanford who are very good at using non-culture and DNA-based technologies, they were able to take samples of amniotic fluid and do both culture-based and non-culture-based analysis on these samples of amniotic fluid. These samples of amniotic fluid ranged from moms who were born, who had their babies born at term, down to very preterm. And some of the microbes they were able to culture, and there were some microbes in that fluid that they were not able to culture, but that they were able to find using the non-culture-based techniques. But of interest is the fact that if you look at the total amount of DNA, the bacterial DNA, the gene copies in the amniotic fluid, so this is as you increase the amount of microbial DNA in the different samples of amniotic fluid, the gestational age goes down. So you've got this inverse relationship. More microbial DNA in amniotic fluid, the greater the degree of prematurity. In these babies, in these amniotic fluid samples over here, they found higher IL-6 and leukocytes. So a greater inflammatory response in that amniotic fluid, suggesting that perhaps these microbes played a role in inducing an inflammatory response. And what's the next step after an inflammatory response? Preterm labor. Okay, very interesting phenomenon. Kirsty Agard from Baylor University looked at 320 placentas and did both metagenomics and 16S sequencing on these 320 placentas and found microbial DNA in almost all of them. She didn't do cultures, but she did the DNA-based technologies. And she found that there was also a relationship between preterm birth and the types of microbes found in the placenta. Work that we did, this is work that one of our fellows did fairly recently, and this is now accepted for publication. It will be published within the next couple of months. We looked at gastric aspirates in babies who were born preterm versus term. And we asked the question, do first gastric aspirates reflect amniotic fluid? And might these differ in term versus preterm infants? So we sort of conjectured that if you get a gastric aspirate within the first hour after birth, this would reflect amniotic fluid. Okay? How do these compare to microbes present in other niches, such as the vagina or the GI tract? Could these microbes relate to inflammation of the GI tract and preterm birth? Well, here's what we found. The samples that have the blue arrows down here are from term samples, and these are preterm samples. These term samples, here you can see, are very different in color than the preterm samples. What do we see primarily in the term samples? We see Lactobacillus genera, okay? In the preterms, we see a lot of ureaplasma of the Tenericutes types of microorganisms. So they're different depending on their level of prematurity. I do have to say that the biomass, the microbial biomass that we found was actually quite low. In many of our samples, it was actually almost at the control levels, but we were very, very careful to look at our control samples to make sure that we weren't measuring what's in the kits or in the background information. But we were able to find microbes primarily that were very different in the term babies that were very different than in the preterm babies. So how do these microbes get into the fetus, and what are the implications? Well, we know that the fetus swallows approximately 150 milliliters per kilo per day of amniotic fluid. And if that happens to be the case, you should be able to find microbes in meconium. And in fact, when we were doing our necrotizing enterocolitis study several years ago, we took meconium samples from preterm babies, and we were surprised that we were actually able to find microbial DNA in these meconium samples. We were also able to culture a lot of microbes from the meconium. And we found that there was about a 50% correlation or relationship to the microbes that were published previously in studies of amniotic fluid. So that suggests that perhaps some of these microbes that we are seeing in the meconium may be swallowed microbes. So meconium is a proxy for the in utero intestinal environment, and we could actually study what is going on in utero in terms of the types of microbes that we see in the meconium. Meconium does not reflect what the baby is eating beyond those first couple of days. It reflects what is going on in the uterus. So I think that meconium is a very interesting body material to study. We did a second study, which was also published, and we found that in babies who are less mature versus more mature, there are different kinds of bacteria. And we also looked at a inflammatory mediator that is used commonly in gastroenterology called S100A12. S100A12 becomes elevated in individuals who have inflammatory bowel disease, and if they have an exacerbation of inflammatory bowel disease. Well, here we have babies born at 24 weeks gestation up to 40 weeks gestation. We saw higher levels of S100A12 in the meconium of the very preterm babies. So this raises an interesting question. Spontaneous preterm labor. Is it possible that perhaps spontaneous preterm labor may actually be triggered not from the mother, not from the placenta or the mother's immune system, but actually from the fetal gastrointestinal tract? The fetal ileum is a highly immunoreactive organ. And with this, the types of microbes may play a role in terms of inducing an inflammatory response that might trigger preterm labor. This is a hypothesis that still needs to be addressed. Now, even if we invoke a sterile womb hypothesis, even if we think that the womb is sterile, microbes in the maternal GI tract probably still play a role in terms of postnatal immune system development. This is a study that was published in Science just a little bit, about a year and a half ago. Very interesting study where they took mice, pregnant germ-free mice, and colonized them with an E. coli HA107. And the interesting thing about this E. coli HA107 is that it only lives about 5 days in the maternal GI tract. Then it dies off. So if the gestational age, or if the period of gestation is 21 days, and if you inoculate the animals at 10 days, at 15 days, this bug dies off, okay? And you don't see it beyond. So then they looked at the development of the immune system of the babies after these babies were born over time. And they looked at two different, this is the outcomes, this is absolute cell number, and this is a certain cell type that is a component of the innate immune system. And they found that those moms who were inoculated, those babies had higher cell counts up to 60 days after birth. So the bugs in the gastrointestinal tract of the mom play a role in terms of the development of the immune system later on. What about some of the metabolic roles? This is a very interesting study that was done by Omri Koren. Omri actually started out as a coral reef scientist, and then he started working with some obstetricians and fetal maternal people who were interested in the microbiome, and this is now considered to be a classic study published in Cell in 2012. The question that was raised by Dr. Koren was, are the microbes different during different stages of pregnancy? Are the stool microbes different during different stages of pregnancy? And so he did genomic technology looking at the stools of many different moms during pregnancy. And then he compared them to the Human Microbiome Project males and females, just a reference group, okay? And here what we see is something called a principal component plot. And what this does essentially is each one of these little circles here represents a microbiome. The sequence of this microbiome is very different than the sequence of this microbiome. So the further apart these little circles are, the greater is the difference in their sequence. So here we see in the HMP males and females, they cluster over here. The first trimester microbes cluster over here. The third trimester microbes cluster up here. So first and third trimester microbes are different, and pregnancy microbes are different than the non-pregnant male situation. Then he did something interesting. He took these microbes here, the first trimester microbes, and put them into germ-free animals. And let these germ-free animals grow up. Nothing happened. Then he took the third trimester microbes, put them into germ-free animals, and when these animals grew up, they had an obese phenotype and a type 2 diabetes phenotype, somewhat similar to what many females undergo during the last trimester of their pregnancy. They put on more fat, and they also have a greater insulin resistance. So the microbes may actually play this particular metabolomic role. Very interesting phenomenon. So we have to think of the microbes during pregnancy, and pregnancy itself as not just the mother and the fetus as a dyad, but we should start thinking more of this grouping, this holobiont as a triad. At least 35% of the metabolites that we see in our bloodstream, including the pregnant women's bloodstream, have a bacterial origin, and most of these microbes come from the gastrointestinal tract. Now let's get on to another topic. What about C-section versus vaginal delivery? What's the C-section rate here in Minneapolis? Anybody know? 28%. Okay, that's a little bit lower than the national average. The national average, I think, is around 34. You get to Canada, and it's maybe low 20s. You get to Scandinavia, low 20s. But then you go to South America, Brazil, Rio de Janeiro, and the private clinics. It's an accident if a baby is born by vaginal delivery. Okay. In other words, they do a lot of C-sections in certain places, probably for reasons that are not medically indicated kinds of reasons. Now, could that potentially cause harm? Well, we know from epidemiologic studies that there's higher odds of all of these problems with C-section delivery. And the question has been raised, is it possible that the mode of delivery may actually be contributing to this and that the microbiome may be responsible? So here's the hypothetical scheme. With vaginal delivery, you have the baby being exposed to microbes, and the baby gets these microbes into the mouth and gets them into the GI tract, and then you have normal development of immunity. This doesn't happen in C-section. And one of the first studies to actually support this was a study that was done by Dr. Dominguez-Bello from NYU, and this was a study that was done in Venezuela in a small number of subjects where they found in this principal component plot that those babies who were born by cesarean section, their fecal microbes looked more like the mother's skin microbes, whereas those born by vaginal delivery, their fecal microbes looked more like the vaginal microbes, supporting the hypothesis that going through the vagina actually makes a difference in terms of the types of microbes that the baby might end up being colonized with. So this led to something very interesting. If we have all these problems in C-section delivered babies, could we potentially remedy this by inoculating our C-section babies with vaginal microbes? And so Dr. Dominguez-Bello and her colleagues did this study, published in Nature Medicine about 2-3 years ago now. The title was Partial Restoration of the Microbiota of Cesarean-Born Infants via Vaginal Microbial Transfer. So what they did in this study, they had 18 dyads, mother-baby dyads. 7 were born by vaginal delivery, 11 by C-section, and those born by C-section were randomized into either being swabbed or not swabbed. The way this worked is that they put a gauze swab into the mother's vagina for about an hour, and if they were born by cesarean section, babies were born by cesarean section, those that were swabbed had that swab placed into the baby's mouth shortly after delivery, and then the face swabbed and the chest swabbed. And question was, did this actually work in terms of inoculation? It did. Here we see the lactobacillus species in the relative abundance in the anus, and here we see skin. So the red here, and here you don't even see any red at all, these are the C-section delivered babies that were not swabbed. And here you have the vaginally delivered babies, you see the microbial content, and here you have the swabbed babies looking a lot like the babies who were born by vaginal delivery. So yes, this did work, okay? But there's a caveat to this, and this caveat was actually raised by the American College of OBGYN in 2016. And I want to quote in red here, "We recommend against the practice of vaginal seeding until better data are available establishing the safety and benefits of this practice." I've been to places, and I don't know if this has happened to you as obstetricians or neonatologists, but there have been families who have seen this on the internet, and they're asking for this to be done. And so it's nice to have this statement to say, no, we don't think that we're not quite ready yet. Why are we not quite ready yet? What if this mom has unrecognized herpes or group B strep or some other illness that could potentially harm the baby? In preterm babies, this could potentially kill the baby. So certainly this is something we don't want to routinely begin to do. We can do something, however, that appears to help out these babies if you want to restore the microbial milieu. And this is a study that was done in Canada by Dr. Megan Azad, and I want to just point you to these circles over here. These are samples from babies, stool samples from babies that are 4 months old. And these are non-breastfed babies, and you can see these colors, which represent different taxa of bacteria. They're still very different if they were born by vaginal delivery or C-section delivery. This is at 4 months. And here are the breastfed babies, and they look very similar. Very small number of subjects. She has repeated this study, and the repeated study has supported this contention. But we do have to think back a little bit, just address this whole area. C-section babies frequently the mothers get antibiotics. They don't get fed with breast milk right after birth. There's a lot of confounding variables that I think we have to be concerned about. And so this is a whole area that I think is still very muddy, and so we should not jump to too many conclusions. Now, what about some other factors that affect the pregnant women's and the neonates' microbiota? Diet, medications, and stress are three that I'd like to just talk about a little bit. Antibiotic use. Intrapartum antibiotic prophylaxis is provided in 30% of pregnancies. The majority of preterms, less than 33 weeks gestation, are treated with ampicillin and gentamicin for at least 48 hours. The average length of treatment of standard of care practice in preterm neonates is between 5 to 7 days. How do these practices affect the developing microbiome, microbial metabolites, resistance factors, and neonatal disease? Well, here's a cartoon that shows that here's the normal situation, balanced microbial community. You have all these normal functions occurring, and here with antibiotics, you get what is called a disbiotic environment that can change the species diversity, alter the TLR signaling, and also cause immune dysregulation. So what about early antibiotics or acid suppressor use? What are we actually doing? So the question here is, do antenatal or postnatal antibiotics affect neonatal microbiota? And this is a study that was done by Arbiola and published about 2 years ago. And they looked at the microbes at a phylum level. And here are the three different taxa or phyla of microbes that represent these different colors. So the first bar here is a mother with no antibiotics and an infant with no antibiotics. And here we see these three different phyla of bacteria. Here is the mother with no antibiotics, but the infant is getting antibiotics. You see the proteobacteria increase fairly significantly. Here's the mother getting antibiotics and the infant no antibiotics, proteobacteria very high. These are primarily gram-negative microorganisms, Klebsiella, E. coli, etc. Mother antibiotics, infant antibiotics, again, largely shift toward the proteobacteria. So yes, antibiotics do affect these, the microbial ecosystem. This is a very interesting study that was published in Nature Medicine by Dr. Deschmuck, who is now at Washington University. And this was done in mice. And the question that was asked was, early antibiotics, if you give antibiotics to a mother, what is the mechanism for late, is there a mechanism that actually causes late onset infections in the baby? So what he did was give antibiotics for 3 days or 5 days versus no antibiotics at all. And here's what was seen. Here's no antibiotics at all in the dark. Here's 3 days and 5 days of 5 days and 3 days of antibiotics. You can see that the microbial DNA in the baby was markedly reduced if the mother got antibiotics. Here is the no antibiotic microbial ecosystem. Here's the antibiotic microbial ecosystem. Very different. And these two graphs here represent development of the certain cell types that relate to the immune system. And you can see a suppression of the immune system with giving these maternal antibiotics for 3 to 5 days. He also saw more infections, actual infections, E. coli with K1 antigen in those animals whose mothers were treated with the antibiotics. So here's from one of my colleagues. So I give a couple days antibiotics to my preterm patients. What does that matter if I change the microbes in the GI tract, since I could potentially be saving the baby's life by treating unrecognized early onset sepsis? Have you asked yourselves that question? Okay. Well, here's what happens to the microbial ecology. Here's week 4 and week 8. Here's the controls. Okay. Compare these colors to the colors up here. They're very different, aren't they? Here's after 8 weeks. Very different still after 8 weeks. So 2 days of antibiotics do affect the microbial ecology of the gastrointestinal tract up to 8 weeks after birth. How does this affect the baby? Well, we know that more antibiotics are associated with increase in necrotizing enterocolitis. So this is one of 3 studies that showed days on antibiotics on the x-axis, and on the y-axis you see the odds ratio for the development of necrotizing enterocolitis. So the average length that we treat our babies in the neonatal intensive care unit, 5 to 8 days, was associated with an increased odds ratio of 50% of necrotizing enterocolitis. In the past, we used to use ranitidine or other H2 blockers very commonly in neonatal intensive care. Why? Why did we use so much ranitidine prior to about 5 years ago? H2 blockers. Apnea, okay? We thought that these babies who were having apnea were having apnea because of their reflux, okay? So a lot of preterm babies prior to about 5 years ago were getting H2 blockers, almost routine in many neonatal ICUs. Well, this particular article says ranitidine is associated with infections, necrotizing enterocolitis, and fatal outcomes in newborns. And this was a multi-trial that was done in Italy. How does this work? Well, here is some evidence that suggests that the microbes are shifted with the use of H2 blockers. During this period of time, this particular baby was getting an IV H2 blocker, and the proteobacteria went up. The firmicutes were low. The H2 blocker was stopped, and the proteobacteria went down, the firmicutes went up. It was restarted again with PO, and the firmicutes went down, proteobacteria went up. And if you come to my lecture at noontime today, I'll show you how proteobacteria may actually relate, or the firmicute to proteobacteria ratio may actually relate to the development of necrotizing enterocolitis. What about TPN? This is a study from University of Michigan, and there are several other studies suggesting something very similar, that adult mice that are fed have lots of firmicutes in their gastrointestinal tract, but if they're on TPN, not having any enteral feeding at all, they have a lot of this blue. These are the proteobacteria, gram-negative, the Klebsiella, the E. coli, etc. Now let's get into breast milk a little bit. And in 2012, the American Academy of Pediatrics put out some guidelines. And these guidelines stated mother's own milk, fresh or frozen, should be the primary diet, and it should be fortified appropriately for the infant born weighing less than 1.5 kilos. So only donor milk or baby's own mother's milk should be used according to these guidelines in our very preterm babies. In many neonatal intensive care units, I know ours went in that direction. I would guess yours did also. And so this actually led to babies being not given formula. This leads to some questions. Donor milk. Where's the microbiome in donor milk, and what are the implications of these AAP recommendations of exclusive human milk for preterm infants for babies of mothers who cannot provide some but not enough of their own milk in the first days after birth? Well, here's some of the things that happen with the pasteurization process. Pasteurization meaning 62.5 degrees centigrade for 1/2 hour. I want to point out here, alkaline phosphatase, almost totally wiped out. Bile salt stimulated lipase, almost totally wiped out. IgM, abolished. Many other factors that are considered to be valuable factors in milk, baby's own mother's milk, are diminished with the use of donor milk. So this is a potential problem. And in addition to these, the microbes in the viable cells are also wiped out. Now, why might this be important? Well, here we have a study from Moscow, Idaho, which was published in PLOS One in 2011. And this is a study that evaluated breast milk from 16 mothers. Each one of these S's represents a different mother. And each one of these bars over here represents a different time. This is 1 week, 2 weeks, and 4 weeks. And you see in this particular mother, the colors or the microbes are the same. In this particular mother, they're pretty much the same over time. But one mother is very different than the other mothers. All of these mothers are very different from, pretty much very different from one another. So this suggests that perhaps the microbes are somewhat personalized. They're different in each particular mom. How many microbes are there in human milk? What is the actual concentration of microbes? This is a study done from Spain, looking at the number of microbes in human milk samples. And here we see, I'm going to shift, the pointer is working, I've got a different one. Here we see that the number of microbes are around 6th in colostrum, transitional milk, and mature milk. These are from the individual mothers. You can see some go very high. So there's actually a fair number of microbes in human milk. Where do they come from? We're not totally sure, but probably from the mother's GI tract. I think that there's more and more evidence suggesting that they may be translocating from the mother's GI tract, or there are certain cell types similar to the dendritic cells, which may be actually picking up these microbes and transporting them through the body to the mother's milk. We have to think about the microbial dose from human milk. If you assume a healthy baby who is drinking 800 milliliters a day, and if you assume 10 to the 5th to 10 to the 6th bacterial cells per milliliter, this will provide 10 to the 7th to 10 to the 8th bacterial cells, which may be personalized daily. And that's very close to the dosage that is used in most probiotic studies. So we are actually giving microbes, microbial therapy, with fresh human milk to our babies, but it can't be donor milk. So one of the things we've started to do is it possible that we might be able to take donor milk and make it better by taking some of the baby's own mother's milk. A lot of mothers in the first several days are not able to produce much of their own milk, but they can produce some. So if you can get a couple milliliters, is it possible to take these couple milliliters of the baby's own mother's milk, put it into donor milk, and refaunate that donor milk to look more like the baby's own mother's milk? And so we did this, and we published this in a paper just last year, Personalization of the Microbiota of Donor Human Milk with Mother's Own Milk. And what we found was that taking a 10% dilution and letting it sit for 4 to 6 hours was actually the ideal to refaunate that milk, to get the donor milk looking a lot like the baby's own mother's milk microbes. So in summary, as far as that study, I'm not going to talk about it, but each mother has a unique milk microbiota, and the live microbiome in donor breast milk can be restored with these unique bacteria using small amounts of the baby's own mother's milk. And as I said before, 10% of the mother's own milk for 4 hours is a reasonable restoration strategy. And I want to say this, don't do this yet. Okay, don't start out doing this, because there are some potential downsides. One of the things we notice is that if you let that milk sit for greater than 8 hours, you see a major overgrowth of certain staph species. Okay, we have to do a lot to be able to do this right yet. So don't start out doing this yet. Future studies should include larger sample sizes, the activity of the microbes in the milk in comparison to donor breast milk and mother's own milk samples, and then clinical evaluation of the safety and efficacy of this practice. Now, I mentioned before that all of this lecture had to do with bacteria. I know that there's some really good work going on here with fungi, but the virome is also very important. And I think that the fungi and the virome of the human gastrointestinal tract also very exciting new areas for exploration. And I also, in the last 50 or so minutes, talked a lot about the taxonomy of microbes, and I talked about the microbiome. But remember, the microbiome does not work by itself. It works in conjunction in the human being with our own DNA, with our nutrition, with various other environmental factors. And so these are working in concert with one another. And we are now developing bioinformatic technologies to look at not just the microbiome, but also the epigenome, the proteome, the metabolome, and also environmental conditions, and putting them together, being able to make heads or tails of different situations. So this is a really exciting area in this microbiome field. So I'd like to stop here with a few take-home messages. Studies of the microbiome are opening new avenues in our understanding of health and disease. The fetal maternal unit most likely is not sterile, and prenatal exposure to microbes may be very important in subsequent development. Early life, pre, peri, and postnatal exposure, such as antibiotic use and diet, are particularly important in that they can result in lifelong changes in health and disease. What mothers and babies eat may not only affect their microbiome, but also have epigenetic effects through production of microbial metabolites. These may not just affect that particular baby through its lifetime, but may also have effects on subsequent generations. Future studies will utilize multi-omic approaches to better delineate how microbes interact with the environment and the host, and the cause or prevention of disease. I'd like to acknowledge many of the people that I've had the opportunity to work with over time, and thank you for your attention. Happy for any questions that you might have.
Questions for Joe? Just beware what fluid you deal with, right? It seems like every fluid has microbial content and potential for affecting life. You know, in the pre-B group, we talked about relevant health outcomes, and you talked a lot about immune priming. Immune health is one of those outcomes. Cardiometabolic health is one of those outcomes. Neurodevelopment is one of those outcomes. Risk of cancer would probably be the other one. Is there any one of those that the microbiome hasn't been associated with, or
There are studies, there's association with all of those factors that are ongoing right now. I think that we are still in a very early phase of learning about the microbiome. It's just like with so many other fields. You have this, exponential increase in interest, and then you begin to find some little holes in the whole area, and then it begins to even off a little bit. And I think that we're starting to slow down, at least in terms of the microbes themselves and naming the microbes, but the metabolomics and the other factors that are associated with the microbes, the things that these microbes actually are doing now are picking a lot of interest.
I'll warn you, we have ID folks out there, GI folks out there, neonatologists out there, surgeons out there. So
yeah, sorry about the antibiotics.
ID guy.
I'm curious to know if there's any correlation between the developing mucosal immunity of the pulmonary mucosal immunity and preterm labor. If the older babies have a better ability to control the microbes that they're exposed to in the amniotic fluid versus a more immature mucosal response.
There are now several studies that look at issues in preterm babies like bronchopulmonary dysplasia. And the microbiota in those babies are very different. And some of the immune factors are very different in those babies who subsequently develop bronchopulmonary dysplasia. So I think that there is an emerging field of knowledge related to the lung and the microbiota. In order to, I can't tell you all the specifics, but the answer to your question is yes, there is a relationship. There's work being done on this. The actual mechanisms, I think it's going to take a little while. We're still in an association phase for a lot of these studies.
Thanks, Joe. Excellent as usual. My question is, we're reluctant to use probiotics in infants, especially preterm, but what about prebiotics, helping to enhance nutrition in the colon, and also would that help beneficial bacteria or not?
So that's a really good question, and I think that there's a lot of work being done on prebiotics. So we have different kinds of prebiotics. We have the plant-based prebiotics, certain oligosaccharides that come from plants, and then we have the HMOs, or human milk oligosaccharides. And there's intense investigation that is now actually going toward the human milk oligosaccharides. The problem is there's about 200 of these guys. And we don't know which are the most important, and which, what kind of a environment that these oligosaccharides actually have to be in to play their most beneficial role. There are certainly studies that suggest that certain human milk oligosaccharides will increase bifidobacteria, certain kinds of bifidobacteria. But whether this actually prevents necrotizing enterocolitis or prevents disease, I think we're still on shaky grounds with those studies. But yes, I think that that's an area that certainly deserves further work. I don't think that we have a product that I would personally consider to be studied well enough yet to say that this really is, the panacea, or this is something that's really going to work.
Other questions for Joe? Will he be around? Oh, Cheryl.
Can you speak to how microbiome research has maybe shaped what you're doing in your newborn intensive care unit now as far as antibiotic stewardship?
Excellent. So fortunately, I was able to, we were able to garner a National Institutes of Health grant to actually do a pragmatic randomized trial of antibiotics in preterm babies. Okay, so what we are doing is putting our preterm babies into 3 different groups. One is a group where moms have very high fever, where they're very, very high risk. pus in the cervix. You know, we can't say we're not going to treat those babies. Those babies automatically get treated. And then we have a group where the babies, there's no risk factors at all. The baby comes out not on a ventilator, having no symptoms. Most of our neonatologists in our unit would not treat those babies. The rest, which constitute about 70%, get randomized into antibiotics or no antibiotics. Okay? And we've enrolled 92 babies as of yesterday in this study. And it's of interest that, you know, one of the things that's happened is that just having that study in our neonatal ICU has started to change the culture. One of the things that has happened is that those babies whose parents do not want their babies in the study, even if they have symptoms, some of our neonatologists are saying, oh, let's just watch this baby. Let's not treat with antibiotics. Whereas 3 years ago, those babies would have automatically been treated with antibiotics. So, yeah, I think that it's slowly beginning to change the culture of our neonatal ICU, but it's sort of a halo effect of this study.
Great, thank you so much. I'd encourage people, if you want to come to the Neonatal Fellows Conference, it's in a smaller room, it's in Wilf 3, but please feel free to stop by and listen to Joe. He'll be there for about an hour and a half today, starting at noon. Joe, thank you for coming. It's wonderful, thanks.
You have been disconnected from the meeting.